Hello everybody, welcome back to another edition of Edpuzzle Lecture Notes. This is going to be the second Edpuzzle on the action potential. It's going to be a very short one, only 4 slides. So here we're going to start with the neuron, this neuron right here, becoming stimulated, and we're going to say it's becoming stimulated by another neuron. Remember, it's going to be, so we're going to zero in on this section right here. One little section of the dendrites. Remember, that would be a multipolar neuron because it's got several processes or appendages branching off from the cell body. Alright, so right here, this is like a close-up view of right there. So down here in the little brown, that's the inside of the neuron, and up here in the blue, that's the outside of the neuron. So if you remember, the resting membrane potential of negative 70 millivolts is kept partially by the sodium-potassium pump, which continuously pumps out 3 sodium ions and pumps in only 2 potassium. So it keeps pumping out 3, pumping in 2. Now remember, it's also kept at negative 70, that resting potential, by the leak channels. More potassium ions are able to leak out then sodium ions are able to leak in. So that's also what helps keep it at negative 70 millivolts on the inside relative to the outside. But now we're going to say an adjacent or nearby neuron has released excitatory neurotransmitters into the space between the two neurons called the synapse. Well, here they are. That's the up-close look of the neurotransmitter. When one of those neurotransmitters fits into this gate, this channel, remember this is a ligand-gated channel because it opens up to a specific key, in this case a neurotransmitter that fits in right there. When that happens, this gate opens up and allows these sodium ions to rush into the cell. Since these sodium, should be doing in red, since these sodium red ions are moving in, the inside of the cell becomes more positive on the inside. Now, remember this area right here. If at the axon hillock, if that number reaches the magic number of negative 55 millivolts, that'll cause a chain reaction of all the sodium, voltage-gated sodium channels to open, and that will cause the action potential to go along the whole length of the axon. Alright, so now we'll see how it kind of works, how it propagates down the neuron. So we started out here at the dendrites where the little neurotransmitters fit into the ligand-gated channel, which allowed sodium to start coming into the neuron. Now, if enough of these ligand-gated channels have been stimulated by excitatory neurotransmitters, at the axon hillock, if it reaches negative 55 millivolts, it's going to spring an action potential, and it happens like this. So now we've zoomed in right here at the axon hillock. So we're gonna assume this is the first of the voltage-gated. Remember, voltage-gated. These guys open and close due to a specific voltage. In this case, it's the negative 55 millivolts. So if enough sodium has come in, because enough of those excitatory neurotransmitters have fit into the ligand-gated channels to allow sodium to come in, if it reaches negative 55, this guy is going to swing open. And that's going to allow sodium ions to rush in to the neuron. And that's going to make it switch from its negative 70 resting potential all the way up to positive 30. Well, that positive 30 is now going to cause this guy right here, right next to him, to open up. That's going to cause more sodium ions to rush in. And it goes down the line. So the sodium coming in right here is going to cause its next neighbor to open up, which causes more sodium to rush into the neuron. And that's how the action potential propagates all the way down the axon. It's just one after the other. One sodium channel opens, which causes the next sodium channel to open, and it does that all the way down to the axon terminals. Now, to return it to its resting membrane potential, When it reaches this positive 30, that triggers these voltage-gated potassium channels to open. And that causes the potassium, these positive ions, to rush out. So this happens right after the action potential has passed by. It causes the potassium to rush out, and then that will also cause this guy to open up, which causes these potassiums to rush out. And that is what causes the neuron to become repolarized. Repolarized. So it goes from positive 30 when the action potential went by, and it goes back to its resting membrane potential of negative 70. Now remember, for a brief moment, it will actually become more negative than negative 70, and that's what's called hyperpolarization. All right, so now we're going to zero in on the very end of the neuron, the synaptic end bulb, the end of the axon terminals. So remember, we started out here with the ligand-gated channels in the dendrites, which receive stimulation by excitatory neurotransmitters, which allowed sodium to begin coming into the cell. If at the axon hillock it reached negative 55, that caused the voltage-gated sodium channels to open, which allowed the action potential to propagate down the neuron. One sodium channel caused the next one to open, and then the next one, and then the next one. It's a chain reaction. And remember, to repolarize the neuron, that's where potassium exited the neuron to take it back. So it reached all the way up to positive 30. Well, with the positive sodiums going out, that's what repolarizes the neuron back to its resting membrane potential of negative 70. Well, now, once the action potential reaches the axon terminals and the synaptic end bulbs, so once these little positive sodiums reach the very, very end, right here at the tip, that causes these voltage-gated calcium channels to open up at the end. And that causes calcium ions to begin rushing into the neuron. Now what that does, it makes these synaptic vesicles, which are basically like little bubbles filled with neurotransmitters, to come up and cleave with the membrane right here, and through exocytosis, it releases its neurotransmitters into the small space. Remember, that's called a synapse. This is the synapse right here. Now these neurotransmitters, just like in our first slide, will migrate across the synapse and fit perfectly in this ligand-gated channel. That is going to open up and allow sodium to come into this neuron. Now, if it reaches negative 55 at the axon hillock, it'll cause an action potential on this postsynaptic neuron. So this was the presynaptic neuron that the action potential traveled down through, and now this neuron is trying to stimulate this neuron, this postsynaptic neuron, to fire an action potential of its own. And now the two types of conduction down the axon. The first one is continuous conduction. Remember, this is where a neuron is unmyelinated. So this neuron does not have the myelin around it that this neuron has. So in this one, it's got to travel the entire length of the neuron. It cannot skip any spaces. So basically it's one channel after the next, sodium channels that open up, and the action potential not slowly, it's still fast, but this one is slower because it has to travel the whole length of the neuron. On the other one, called saltatory conduction, that's when the neuron is wrapped in myelin, either by a Schwann cell in the peripheral nervous system or by oligodendrocytes in the central nervous system. This is where it's the same thing, except due to the myelin, this impulse is able to jump from node of ROM-VA to node of ROM-VA. So that makes it much, much faster. So you can see after 10 milliseconds, this neuron is almost done. It's traveled about 3/4 of the way down the axon. Well, after 10 milliseconds in continuous conduction, this guy's only traveled about 1/3 of the way down. So what you need to know, saltatory conduction is because the axon is enclosed or covered with myelin, and this is much faster than continuous conduction. All right, thanks for listening. I hope this makes it a little bit easier to understand. and I'll talk to you next time.